Okay, welcome to Test Prep for Chapter 10. This chapter is all about the processes of sexual reproduction, and therefore about genetics, which explains not only the mechanics of forming gametes, but the ability to predict inheritance patterns. We'll start with a discussion of the two types of cellular division we've discussed in class, mitosis and meiosis. Let's compare and contrast mitosis and meiosis. The DNA content of the resulting cells in mitosis is a diploid content, 2 copies of each chromosome. Haploid in meiosis, 1 copy of each chromosome. The structures we follow in mitosis are chromosomes and chromatids, while they are tetrads in meiosis, 4 copies of the chromosome together. Crossing over does not occur in mitosis, but it does occur in meiosis, mixing and matching genetic information. The resulting number of cells, 2 cells in mitosis, while we get 4 gametes, or sex cells, in meiosis. Relationship of those new cells to the original are, in mitosis, they are identical, while in meiosis, they are all 4 different. Types of cells that do this: normal body cells do mitosis, almost all cells in the body, while only gametes, or sex cells, in the sex organs do meiosis. Location it occurs almost everywhere in mitosis, but again, only in the sex organs for meiosis. Okay, the next thing is going to be important for you to be able to recognize is the possible results of the cell division with regards to where the chromosomes end up. Let's start with a cell from an imaginary organism that only has 2 pairs of chromosomes. The starting cell, no matter which division we are discussing, is diploid. That means it will have 2 copies of each chromosome. In this case, it's a long one and a short one. The shading indicates if the copy was from this organism's mother, light-colored, or from its father, dark-colored. If we look at cell A, we see 1 copy of each chromosome, 1 short and 1 long. This is the haploid cells that would be produced from a meiotic division, a gamete or sex cell. Next, cell B has 2 copies of the long chromosome and 2 copies of the short chromosome. Even though they're not in the same physical location as the original cell, these 2 cells are genetically identical. Therefore, this would be the product of a mitotic division. Cell C technically also has the same amount of DNA as the original cell, but notice they are copies of each other and they are joined together. This means that these are really 1/2 of tetrads, 2 sister chromatids joined by a centromere. Therefore, this cell must be in prophase 2 of meiosis. Now, cell D has 2 copies of each chromosome. If this is prophase of mitosis, it would next proceed to metaphase after forming chromosomes. But it's also possible that this is prophase 1 of meiosis, and that the next step would be for those structures to form tetrads. In any event, the doubling of chromosomes indicates that the cell has gone through S, and depending on what forms next, it was either prophase of mitosis or prophase 1 of meiosis. Okay, so let's talk about a few other things that will be on the test. Always keep in mind that the test is about meiosis. It's about the process of meiosis and the ability to use that information to explain and predict inheritance patterns. The key feature of meiosis is that cells duplicate their DNA once, but divide the cell twice. This means that only possible time you would ever see a cell dividing without duplicating its DNA would be in meiosis 2. Recall that crossing over is the ever-important exchange of genetic information between homologous chromosomes and happens during prophase 1 of meiosis. This greatly contributes to the genetic variability of gametes. This process ensures that only the original maternal and paternal chromosomes are not passed on, but rather new chromosomes with some information from each. Also recall that when a sperm and egg combine, or any male-female gametes combine, fertilization occurs, leading to the formation of a stem cell for a new organism. Remember that gametes have 1 copy of each homologous chromosome, so they have half as many as most cells, making them haploid. Since humans have 46 chromosomes, each sperm and egg formed should only have 23 chromosomes, 1 of each homologous pair. Lastly, you will need to be able to do simple Punnett squares. They will be monohybrid crosses, so one trait with one gene controlling it and two alleles in the population, dominant and recessive. You need to be able to fill out the Punnett square and predict the ratio of offspring. Again, I'm going to tell you to pay close attention to this review and the formative assessments in the chapter, and you should be all set for the test.